Good afternoon and happy Friday, everybody, from beautiful Cape Town here in South Africa. I want to thank everybody that's joined our webinar today to listen to an amazing, it's going to be an amazing talk given by Dr. Riyad Domingo, our newly appointed Chief Innovation Officer at M2 Biosciences. It's been an incredible journey getting to know Riyad and how his brain works and what he thinks, and how we're very much similar in our approach of trying to not always follow what everybody else is doing, but having a contrarian viewpoint and looking at different methods to bring in this case, medicine to the forefront, using things like artificial intelligence and machine learning. So without, butchering anything that Dr. Riyad is going to talk about, I'm going to directly hand it over to you, Riyad. You're the doctor with all the incredible knowledge here, and I'm looking so forward to hearing what you have to say. Over to you without further ado.
Thanks, Jeff. Good afternoon, and thank you for joining us today. I'm Dr. Riyad Domingo, the Chief Innovation Officer for M2 Biosciences. In today's webinar, I'll be providing a brief overview of the psychedelic psilocybin and discussing the potential of this compound in the future of mental health. So just a little bit more about me. I completed my PhD in medical biochemistry at the Institute of Infectious Diseases and Molecular Medicine in collaboration with the Drug Discovery and Development Center, or H3D, at the University of Cape Town. My research dissertation focused on one of the primary therapeutic targets of hypertension, angiotensin converting enzyme, or ACE. For those unfamiliar, the homologue of ACE, given the very uninspired name ACE2, serves as the entry point for SARS-CoV-2 coronavirus infection, the virus causing COVID-19. My past areas of interest have included cardiovascular disease, hypertension, sports medicine, and antibiotic-resistant bacteria. Upon concluding my academic career, I co-founded a medical device digital manufacturing startup, and my team and I were very fortunate to be featured on TEDx, Brightlands, and were most recently selected as one of the most innovative companies in South Africa by Fast Company SA. I've joined M2 Biosciences as the Chief Innovation Officer, and I will be leading them through the next part of their growth as they explore known and novel therapeutics in the future of mental illness. M2 Biosciences is one of the leading medicinal CBD companies in the world. The subsidiary, MJ Medtech, is a nutraceutical biotechnology company that researches, develops, and commercializes a range of CBD-based products under the Dr. Anna, Medspresso, and Liviana brands. You can also see our vision, mission, and core values here. They're also available on our website. So just before I begin today with what we believe could be the future of mental illness therapeutics, I'd like to define the scope of this presentation. I will not be focusing on anecdotal reports or subjective evidence on the effects of psilocybin. Anecdotal evidence is usually based on individual experiences or observations. So I will not focus on mysticism and other related practices. It is also important to note that psilocybin and psilocin, the psychoactive substance, are listed in Schedule 1 of the Controlled Substances Act in the US, making it illegal to cultivate or possess psilocybin-producing mushrooms for either personal consumption or distribution. So today I'll be focusing on the molecular and pharmacological effects of psilocybin and the potential for use as a clinical therapeutic treatment. But why is it important that we explore new compounds and therapies for mental health? The medical definition of an illness is poor health resulting from disease of body or mind. A mental illness significantly affects how a person feels, thinks, behaves, and interacts with other people. Mental illness includes a range of conditions for which there are standard criteria used to diagnose them, such as major depression, PTSD, bipolar and eating disorders, anxiety, substance use disorders, and schizophrenia, Depression, one of the most common forms of mental illness, is characterized by sadness, loss of interest or pleasure, feelings of guilt or low self-worth, disturbed sleep or appetite, tiredness, and poor concentration. People with depression may have multiple physical complaints with no apparent physical cause. Depression can be long-lasting or recurrent, substantially impairing someone's ability to function at work or at school or just to cope with daily life. At its most severe, depression can lead to suicide. In 1990, the WHO projected that depression would increase from fourth to the second most frequent cause of worldwide disability by 2020. In 2008, it was ranked third and projected to be the first leading cause of disability by 2030. If you have any signs or symptoms of mental illness, please see your primary healthcare provider or a mental health professional. Most mental illnesses don't improve on their own, and if untreated, a mental illness may get worse over time and cause significant problems. But it's actually more common than you think. Mental and substance use disorders are common globally, as indicated by these data from the Institute for Health Metrics and Evaluation, as reported in their Global Burden of Disease study in 2017, prior to the global pandemic. In this map, we can see that globally, mental and substance use disorders, including the ones that I mentioned prior, affect around 1 in 7 people. That's 15%. That would be over 1 billion people around the world that are affected by mental illness. It is important to note that many public health experts actually believe that these estimates are a gross underrepresentation of the true burden of mental illness, especially in lower income populations and in developing countries. In this graph from the same study, we can see that anxiety disorders lead the burden of disease, followed closely by depression, then alcohol and drug use, substance use disorders, followed by bipolar disorder. So it really is quite common. So now moving more onto the molecular basis. So serotonin is one of the most important monoamine neurotransmitters in the body and has a complex yet versatile role in regulating mood, cognition, sleep, reward, learning, memory, and it plays a role in numerous other physiological processes, including digestion. Serotonin is secreted by a presynaptic neuron and binds to receptors on a postsynaptic neuron, most notably the 5-HT2A receptor. Serotonin levels in the synaptic gap fall partly because reuptake transporters take back available neurotransmitter, in this case serotonin, into the presynaptic neuron. The monoamine hypothesis of depression predicts that the underlying pathology of depression is caused by a depletion in the levels of available serotonin and other neurotransmitters in the central nervous system. And one of the most important, one of the main first-line treatments of depression is the serotonin reuptake inhibitors, or SSRIs. In depression, as I mentioned previously, we have, in this second figure, we have fewer monoamine neurotransmitter molecules available for binding to receptors on the postsynaptic neurons. Treatment with serotonin reuptake inhibitors, in this instance, this red square, symbolizing Prozac, one of the most common SSRIs, blocks the activity of the reuptake transporters. thereby increasing the number of neurotransmitters, or in this case, serotonin, available for binding onto that postsynaptic neuron and eliciting a response. This usually corrects the mood disorder, but why do I use the word usually? Although these drugs have enjoyed some success over the years and are able to induce remission, approximately 1/3 of patients show no response to any class, while other patients respond only partially. 100 million of those diagnosed with major depressive disorder are said to suffer from treatment-resistant depression, or TRD. An individual is diagnosed with TRD after they are observed to have failed to sufficiently respond to a course of antidepressants within a set and specific period. There are also concerns about the market dominance exhibited by these antidepressants, as these compounds come with a number of safety concerns. And these side effects that are associated include insomnia, drowsiness, and most concerningly, an increase in suicidal ideation. The global antidepressant market was valued around 13.7 billion in 2016 and is projected to grow to just under 16 billion by 2023. Because of this, new therapeutic approaches are being researched and aggressively pursued. In this slide, we can see the structures of serotonin, or 5-hydroxytryptamine, number 3 in this diagram, and other related tryptamines, including the hormone melatonin 4, which plays an extremely important role in the natural sleep-wake cycle. However, of particular importance to us here at M2 Bio is the prodrug or precursor psilocybin, designated 1, and the active psychoactive substance psilocin, labeled 2. To date, of all the naturally occurring psychedelic alkaloids for mental illness, research on the potential of psilocybin has been the most promising. Studies have demonstrated a significant decrease in depression and anxiety symptoms in patients with moderate to severe TRD. And effects were sustained from 1 week to at least 6 months after a single treatment. And of all psychedelic drugs, psilocybin is reported to have the most favorable safety profile. Evidence-based data suggests that psilocybin may be the most efficacious psychedelic drug for treating mental illness. It's no wonder it's been labeled as breakthrough therapy for major depressive disorder by the US FDA in 2018, and has also been approved for phase 3 clinical trials by the European Medicines Agency. Some current psilocybin-assisted therapies being explored include alcohol, stimulant, cocaine, tobacco, nicotine, and opioid addiction, cannabis dependence, as well as treating a number of anxiety disorders, including PTSD, OCD, advanced stage cancer-related anxiety, and cancer-related depression. As I mentioned previously, we see TRD and major depressive disorder. Interestingly enough, we also see inflammation, and that is something that we at M2 Biosciences have contributed to. And I'll speak more about that in a little bit. So, the structure of psilocybin was first elucidated by Albert Hofmann, seen here, a Swiss chemist considered the grandfather of psychedelics, who would often test these substances on himself. He purified psilocybin from a psilocybe mushroom species found in Mexico in 1958, 1959. Here we can see the biosynthetic process used by the mushroom species. It has only recently been identified And we can see that it usually starts with the naturally occurring amino acid tryptophan. The reaction proceeds through a number of enzymatic steps to produce psilocybin. Psilocybin is a prodrug or precursor. But that's great. What happens next? What happens after ingestion? Well, once psilocybin is ingested, it is absorbed and undergoes hepatic first pass metabolism, where it is rapidly converted into the active psilocin metabolite. At this stage, we still do not know exactly how the prodrug psilocybin is converted and processed into the active psilocin. Psilocin then enters the systemic circulation, crosses into the brain, where it exerts its effects by binding to serotonin receptors. As I mentioned previously, the 2A, most notably the 2A receptor, but it does also bind to a number of other receptors. While the psychoactive properties of psilocin are thought to be mainly due to the binding at the 5-HT2A receptor, the pattern of activation of the broad spectrum of receptors and receptor subtypes is responsible for the unique psychedelic profile seen in users of psilocybin. It is also noteworthy to mention that in animal models, psilocybin and psilocin are dosed in a range of between 0.25 and 10 mg per kilogram for behavioral studies, with a lethal dose of 293 milligrams per kilogram. And this gives us some insight as to the wide therapeutic window with which researchers can explore. Into Biosciences is currently working to complete research on the efficacy of psilocybin in the treatment of various conditions, including addiction, mental illness, and cardiovascular disease. The global pandemic has delayed our rather ambitious R&D pipeline somewhat, but we have been making great progress in moving forward. And most recently, we have collaborated with Sana Nkadimeng at the phytomedicine program at the University of Pretoria. In our first collaborative study, we demonstrated that extracts of the Psilocybin thalalensis, a species of psilocybin-containing mushroom found in South Africa, was able to exert a dose-dependent analgesic, antioxidant, and anti-inflammatory effects in lipopolysaccharide or LPS stimulated macrophages. For those unfamiliar, LPS is a major component of the outer membrane of gram-negative bacteria. Thus, these conditions mimic bacterial infection in vitro. This was demonstrated by the inhibition of nitric oxide and prostaglandin E2 production, two inflammatory mediators involved in the pathogenesis of inflammatory diseases. Furthermore, the results proposed that extracts lowered LPS-induced TNF-alpha or tumor necrosis factor alpha and inhibited induced pro-inflammatory cytokine interleukin-1 beta. Both are well-known pro-inflammatory cytokines reported to be in high concentration in chronic inflammatory conditions. Reduction of these cytokines are usually associated with improved prognosis. In another one of our publications, accepted into the Nature Publishing Group's scientific reports, we evaluated the effect extracts of the psilocybin and another species had on cardiomyocytes treated with endothelial 1 to mimic pathological hypertrophy, and finally with TNF-alpha to induce cell injury. These results proposed that the psilocybin-containing extracts did not aggravate the pathological hypertrophy induced by endothelial 1 and also protected against TNF-alpha-induced injury and cell death. And finally, the last research paper was published just last month in the Journal of Inflammation Research and evaluated the effects of 4 psilocybin-containing mushroom extracts on pro-inflammatory mediators COX-2, 15-LOX, and a number of other pro-inflammatory cytokines. The research found poor 15-LOX inhibition, but inhibited LPS, induced production of pro-inflammatory mediators. TNF-alpha and interleukin-1 beta significantly, and also lowered IL-6 and COX-2 concentrations in treated human macrophage cells. So, until now, I've spoken at length about the enormous burden of disease. I've spoken about the molecular potential of psilocybin in treating not just mental health illnesses, but also in inflammation. And I've spoken about where we have been and demonstrated our commitment to furthering the body of research, development, validation, and safety studies of psilocybin in vitro treatments of known and new targets in disease. Now I want to talk about the exciting part. I want to talk about where we are going and the role of M2 Biosciences in the future of therapeutics. And I've simplified these into 3 basic workflow revenue generating streams. These include the synthesis and large-scale production of psilocybin, the development of novel and patentable tryptamine candidates, and finally, novel therapeutic methods of delivery, which leads me into the area of personalized medicine. So one major area of focus for the psychedelic industry is the ability to sustainably produce large quantities of the active pharmaceutical ingredient psilocybin. Our research partner at the University of Pretoria possesses the license to purchase and handle psilocybin for research and development purposes in Africa. And we are also working with the government of Lesotho to develop a framework for the industry. However, purifying a pharmaceutical grade product from natural sources is often problematic due to inefficiency with low yields, expensive to scale, and there are also challenges with ensuring that good manufacturing practice is standardized across species of psilocybin and even across batches and harvests of the same species. The synthetic process for producing psilocybin was developed by Albert Hofmann and colleagues again in 1958, 1959, and it's shown on the left-hand side here. It has remained relatively the same with a few modifications to improve yields along the way. This method, while innovative at the time, is limited in the scope of molecules one may explore, as well as being dependent on non-renewable resources, and also is notorious for producing large amounts of hazardous waste. One of the promising areas that we at M2 Biosciences have begun to explore is the biocatalytic production of psilocybin and other potential tryptamines of interest. Biocatalysis is defined as the use of biomolecules such as enzymes or even whole cells to complete chemical reactions. Biocatalysis often goes hand in hand with the field of green chemistry. Green chemistry is the design of products and processes that reduce or eliminate the use or generation of hazardous substances, and is always based on the 12 principles of green chemistry. Without giving too much away, for our work, we have begun to evaluate the one-pot biocatalytic production of psilocybin at scale by leveraging our expertise in the area. Simply put, we are attempting to mimic the natural enzymatic process that occurs within the psilocybe mushroom species by utilizing modified and optimized enzymes, cheap and renewable starting materials, and of course, reducing our organic waste. We will share these findings at an upcoming webinar soon. Moving on to our second focus. Most recently, the high resolution active state structure of the 5-HT2A receptor with bound prototypical hallucinogen as well as LSD was resolved. This represents a major shift in exploring the use of structure-activity relationships in the area of psychedelic research, allowing us to explore the potential of creating novel and more selective lead compounds that may be better mental health therapeutics by affording us the opportunity to explore in this instance, as we can see with the hallucinogen and LSD, by allowing us to explore differences in ligand binding and looking at a few other characteristics as well. We believe that the future of mental health therapeutics lies with proven and established drug discovery and development approaches. M2 Biosciences has gained a competitive advantage in the industry of psychedelic research by starting to work with one of the largest automated drug design and development platforms in the world. We will be taking full advantage of the power of artificial intelligence and machine learning algorithms to enhance the drug discovery and development process in our search for novel and defendable lead compounds based on the tryptamine class of compounds with improved efficacy and reduction in some of the side effects associated. Through this collaboration, we will also identify key characteristics of compounds that will allow us to create unique and customized sets of drug-like molecular libraries for known and new disease targets. We are also working to expand our partnerships with experts in the field to bring our leads from the lab to life. And finally, we at M2 Biosciences are exploring personalized medicine with regard to psilocybin and other tryptamine derivatives. But it's first necessary for me to explain the entourage effect hypothesis of psychedelics. This is the idea that the sum of the contributing parts of a natural product produce a greater synergistic effect when compared to the active pharmaceutical ingredient alone. While psilocybin is the most abundant tryptamine in the psilocybe species, other related tryptamine derivatives have been identified, indicated here by Sherwood et al. It is highly likely that psilocybin may not act by itself, but in concert with these derivatives or secondary metabolites to exert the physiological effects that we observed. As mentioned previously, there would be significantly different concentrations of these tryptamine derivatives, not just across species of mushroom, but also across batches and harvests if we dealt with mushroom extracts. But I've already mentioned how we are planning to use biocatalysis to circumvent this challenge. But one of our most exciting avenues of research is the applications of pharmaceutical 3D printing in this aspect. By leveraging our experience in 3D printing and the medical device industry, we can finally realize the full benefits of 3D printing in a personalized manner, able to create potential mental illness therapies by combining a number of related yet distinct tryptamines of interest, as mentioned previously with regard to the entourage effect. But also, we would have the benefit of things like dosage specific parameters. We could look at unique forms of delivery. We can look at increased or decreased solubility and the ability to limit processing. We could also expedite our route to clinical trials, and most important of all, we can demonstrate repeatability in the industry. So in exploring these projects, one of the most important things that we wanted to convey is that we are not only looking to create value for employees, investors, and shareholders, but our work hopes to improve the quality of life for patients and their beneficiaries around the world for the future. Thank you.
Thank you, Dr. Domingo. It was great to finally get through it. And I want to thank everybody that was on the webinar that were listening in. Thank you for their patience. It's a fascinating topic, to say the least. We do have a couple questions. One question from David out of Sarasota, Florida. What kind of IP issues are there around the compound psilocybin that is a natural compound available to everyone? Any IP issues?
Well, I mean, the IP issues mean that no one can patent psilocybin, right? The patent has expired. So it's a natural product. It's available for, we hope that, it passes FDA approval and it's granted, a license to be used. Then generic manufacturers can pick it up and manufacture it. That is one aspect. So psilocybin in itself can be a generic if granted approval and can be used to treat various conditions, which is great in one part. But there's also this drive to look at psilocybin as the scaffold to create new compounds that people can defend and take out a patent for, and be able to offer it as an alternative to generic psilocybin for a particular condition. So I'm hoping that answers the question.
So without having to reveal too much, I'm guessing that is what you were alluding to in your presentation when you mentioned that is where we're heading.
So we've got a bit of both. So we spoke at length, or I rushed through it a little bit, but we were looking at using structure-activity relationships and artificial intelligence in helping us improve psilocybin to generate new leads. There's that one aspect where we can generate new compounds that perhaps may be more efficacious than psilocybin itself, and look at taking those through the clinical trials. Second to that, and it was the last part, we can also look at how we formulate psilocybin. Can we look at, for instance, a sublingual, maybe I'm saying too much, a sublingual dose? As we go through your psilocybin-assisted therapy, we know, for instance, that psilocybin usually has a 10 to 40 minute onset, right? You can't really wait 10 to 40 minutes when you're in your session with a psychologist or a psychiatrist. We may look at using sublingual, easily dissolvable psilocybin at a specific dose. So that would be available immediately. So the moment you start your session, your psilocybin can kick in, and the assisted therapy can begin.
Great. And another question, could you explain the relationship between tryptophan, serotonin, and psilocybin?
So tryptophan is a naturally occurring amino acid. I've joked before. I said tryptophan occurs in turkey, which is, we all often eat it on a Sunday, and straight afterwards we fall asleep. But in that biosynthetic process, we can see that tryptophan serves as the starting material for the creation of serotonin and melatonin. And melatonin is involved in the sleep-wake cycle. In humans, we don't possess the enzymes needed to make the endogenous. We don't have the endogenous enzymes to produce psilocybin itself. We can make serotonin, we can make melatonin, we can make a whole host of other things, but we cannot make psilocybin. Give tryptophan, the amino acid, to a psilocybin mushroom, and it can make psilocybin. So tryptophan, the amino acid, serves as the starting material for the production of various neurotransmitters. I often regard it as the amino acid that is essential for a healthy mind. One can get 5-HTP on the shelves. If one is flying to different time zones, we usually get melatonin. They're all derived from tryptophan. I'm hoping that answers that question.
Sure. So good question here. Somebody's asking, I won't mention the name of the companies, but somebody is saying they're a shareholder in another psychedelics company, and they want to know what the difference is between what it is that we're doing compared to these two other publicly traded companies. They are large capitalized companies, and they want to know, what is the difference? What's M2 Bio's differentiator between what we're doing and what they're doing?
You want to take this one, Jeff, or should I?
Yeah, well, I mean, that's actually interesting. I mean, I have my very, I started out the webinar when we first came on. I did say that, there was a reason why we've never participated in many of these webinars, online webinars that have been out there for a couple of years. And this whole space came out of nowhere very, very quickly. We were one of the very first players in it. And I do believe we're one of the very few players that actually have conducted clinical research. So we're very proud of what we've done at M2 Bio and where we're heading. And I always felt it was important that, we only come to the marketplace when we're ready to share a story that has true science, rigorous science behind it. And, from my perspective, I'm not a doctor, of course, and I'm not a brilliant scientist like so many in this field, but I have seen, much like in the cannabis field, a lot of kind of stories about, being in the forest and going out and getting stoned. And that's kind of what I heard a lot of. And that's not who we are at all. And that's why I never wanted to be part of those types of online webinars or whatever they may be. So my thing has always been, when we do these presentations, and this is our very first one with Dr. Domingo, and we will have further presentations coming up very soon, that we deliver real science. And I think that we've done that today. I think that we've delivered something very new. I mean, there is obviously so much more behind what Dr. Domingo has talked about today, but what his approach is in conjunction with what we've done already is extremely powerful. It is extremely new. It is an approach that I have yet to see anywhere. I mean, I'm sure it's out there somewhere maybe, but we haven't seen it. And from that standpoint, I'll just lay it off to you, Riyad, with you can maybe finish up that with a 1 minute, what is that big difference scientifically, how we are approaching what it is that we're doing. I mean, it is just so different. We're not doing the other stuff that's out there. We're actually, many of the companies are just kind of buying science that's been done for a while, and they're saying that it's theirs and this and that, and we're doing this and doing that, or we plan to do this and we plan to do that. That's never been who we are. We have conducted much, and we're going to conduct much, much more. And maybe I'll leave it to you for sum up that in the scientific side.
Yeah, I think you summed it up quite well. What we're doing is we're basing a lot of what we do on evidence-based research. We're looking at what the research says. We're looking at processes that have been used for decades in other industries. And we're taking those lessons, those processes, those approaches, and we're applying it to an industry that, as an outsider, has perhaps lacked a little bit of scientific backing. You know, looking at the for instance, and I had to rush through it, is we're the first group that's looking at, together with the University of Pretoria, we're looking at the role of psilocybin in inflammation. So that has major effects and for us and for the industry. And it just adds another potential application of psilocybin in the future of mental or therapies.
Cool. Somebody's asking, when are the new psilocybin research studies being published? I can answer that. There are 3 more. You never know the answer to that. It's not up to us. It's up to the peer-reviewed board that are reviewing the papers. When they make their decision is when they make it. Generally speaking, I can say it takes 2 to 3 months in between papers being accepted. But having said that, during this pandemic, we found that the last paper actually took 8 months or whatever to be approved. So it's out of our hands. We know that the next 3 papers are very powerful, and we're very proud of that work. So we look forward to having those out there. I think that's it for all the questions I have at the moment. Hang on. Yeah, I think we're done with the questions. And I want to thank everyone again for their patience. It was our first one, and we'll make sure that we improve on the Wi-Fi connection for next time. But the information, I think, was very, very interesting. Feel free to reach out to Dr. Domingo, myself, with any questions. There will be a recording of this webinar available very soon. And we look forward, as Dr. Domingo said earlier in the webinar, that he will be sharing much more information as it relates to the implementation of artificial intelligence and machine learning into our drug discovery process. I can tell you it's a whole different world. I mean, and I'll tell you one little anecdote which I found amazing. And here you have a brilliant doctor. And by the way, Dr. Domingo is a Harvard fellow this year, and I think that's a tremendous accomplishment, needless to say. So congratulations on that. And when we were looking at how the AI and ML can help speed up that drug discovery process, and he said, my God, he says, what took me 8 years of hard work and Excel sheets and spreadsheets every which way all over my desk and 18-hour days, I can now do in 2 minutes. And that just shows you the power of technology and how we can leverage technology to bring, as Dr. Domingo said, precision technology, precision medicine to the people, and give people what they actually need, instead of just giving people what the pharmaceutical companies say. There's 5 milligrams or 10 milligrams, but what about something in the middle? And that something in the middle is where Dr. Domingo alluded to on the 3D printing side, where we could do just-in-time drug manufacturing and stuff like that. It's going to be so exciting. The future of medicine has never looked more promising, and we hope to be a big, big part of that at M2 Bio and deliver some incredible things for everybody. So thank you again, everybody, for your time and patience. We will have many more of these coming up. Dr. Domingo, thank you so much. And thank you, Luca, behind the scenes, wherever you are, who put this whole thing together. He's behind the scenes somewhere. And I wish everybody a fantastic weekend, and we will catch up soon. Thank you so much.
Thank you. Bye.
Thank you.